Nishikawa. Again, I would like to request Manish Biyani sir for the floral welcome of Dr. Vimal sir. Thank you, sir.
Thank you. After the welcome of the dignitaries. Maiko, there are any questions to Maiko Okazima on her topic, on her lecture? Are there any questions? It was a very interesting talk, by the way, because saccharin is a new polysaccharide. In fact, it is origin of Japan, and it has got a fantastic property. It's the highest molecular weight polysaccharide ever found for any other polysaccharide. Saccharin is the most important polymer. No questions? Yeah, very good. Okay.
Mike? One mic.
My question is, what is the significance of thick films? In saccharin, we say that thick films are bacterium thin films. That's all why.
So importance of the thin film? Importance of the thin film?
Ma'am, you are not audible.
It's a difficult question, but when we apply the material for the especially in vivo, so the thickness is important, and if you can imagine if the thick film you put in our body, so it's not good. So looks like our skin. The film looks like our skin, and in our body, hydrogels are like the film. So absorb our body something water, and then if the film thickness is originally thin, so after swelling the water in our body, so it makes no problem. So thickness is important like this.
Thank you, ma'am.
Thank you.
Thank you. I have one question, just a quick question. You showed a very interesting self-healing behavior of two pieces of how much time it takes for self-healing? Time. How long you usually?
We put the hydrogel at room temperature 1 day.
1 day. Okay. Thank you. Thank you very much. Thank you, for the excellent talk on the polysaccharide sampling. Our next speaker is Dr. P.D. Charan, who is assistant professor of Maharaja Ganga Singh University, Biyani, India. Dr. Charan has been working as an associate professor, assistant professor in the Department of Environmental Science at Maharaja Ganga Singh University, Biyani, for the last 5 years. The area of interest is biological diversity and environmental impact assessment. He has 40 international publications, and is also the project leader on the biodiversity from Mandala Oil Field at Bogat Terminal. He has been awarded the Young Geographer Award of 2009. Thank you very much, Dr. Charan, and I invite him to give a talk. Yes.
before going to present my work, I would like to admit here that when I got an opportunity to assess some of the posters outside, and I came to know that the students of this organization are working on some depth check to assess the heavy metals in different samples. So it is very new for me. I have heard about this, but the people are working on this technique. So hats off to your organization that it is doing work in great. I will present my work on the traditional technique. I have used the traditional technique to assess some heavy metals in different vegetable samples. So, from where this idea came into my mind that we have to assess the heavy metals in the wastewater and vegetables basically. So, when we assess the census of India, the last two census of India, we can see that the urbanization in India is increasing at very fast rate. You can see that the red colors in this map shows that more than 35% of the population living in the urban areas. So I took this problem, and the population of Bikaner city is increasing in fast rate from since 1931 to the projected population is shown in this map. You can see this graph 2021. So population increasing at very fast rate, and we have to meet the need of these people from vegetable point of view and all from the same land. So different kinds of pesticides and fertilizers we are using day by day, and wastewater is also generating as far at a large quantity that is being used to irrigate the vegetables also, the trees also. And this is the map of the rainfall pattern of Rajasthan. It shows that the Bikaner lies in the area where the annual rainfall is around 300 millimeter per year. So we are dependent on the wastewater to irrigate our vegetables. If I talk about the industrial area of the Bikaner, you can see that there are three major industrial areas of Bikaner: Karninagar, Bichwar and Rani Bazar industrial area, and around 700 or to 800 industrial units are installed in the city. A huge amount of the wastewater per day they are releasing. So this wastewater may contain different kinds of heavy metals, and if we irrigate the vegetable crop with this water, that may create severe health related issues to human beings. So what kind of the research work which is going on in our laboratory? We are working on the assessment of the heavy metals in different samples. We are assessing heavy metals in ambient air also, in the water and soil also, and in edible commodities, especially vegetables, we are assessing the heavy metals in these vegetables also. So, and some scats of the selected herbivores are also assessed in our lab, and some excreta of top consumers. You can see the photographs here. The last photograph, you can see the vultures. These vultures, you can't see such kind of the huge population anywhere in the Rajasthan except Bikaner. Bikaner is there is one village, Dodvi, which is the site for the dumping of the dead organisms. Thousands of vultures you will see there, and so you can see that from grasses to the herbivores to the top consumers, we are assessing the heavy metals in entire food chain. And in present investigation, we have assessed some vegetables. I took vegetables because vegetables are the the commodity which we are eating at least twice a day. And India is the second largest country from vegetable production point of view after China, and it shares around 3% of the total cultivation of the crops. And vegetable cultivation shares 14% of the total pesticide consumption in the Indian agrarian system. So these pesticides again have different kind of the heavy metals. So we took this problem first. Then you can see in the photograph, some cattle egrets are sitting in this field. This shows that the wastewater is being used for irrigation. You know, the wastewater containing different kind of the worms. These birds are eating those worms. So the wastewater is important source of irrigation for the vegetable production. We have collected samples from 4 agricultural locations from Jorbi, Vallabh Garden, Patel Nagar and Shivbadi area of Bikaner city. At each sampling site, vegetable samples were collected from 3 different locations to provide replicate samples of each vegetables. and we have selected 5 different vegetables, and these vegetables were selected on the basis of the area of the cultivation. So these five vegetables were studied. In 2014 we have studied around 120 samples of these vegetables, and we assessed 4 major heavy metals: iron, copper, zinc and manganese. So some people may ask that why you have selected only 4 heavy metals. See, in the Bikaner region, Bikaner is the largest woolen industry there is in the Bikaner, and for giving different kind of the shades, the colors to the woolen textile, they are using different kind of dyes, and important dyes which are being used having these 4 major heavy metals. So that's why we have selected these heavy metals. These are the methods through which we extracted the heavy metals from the samples, and these are the basic characters of these heavy metals. As the students of the MSc and BSc might know, that the heavy metals are those metals which are having density more than 5 gram per centimeter cube. So only those metals may be categorized under the heavy metal category. So these are the heavy metals having this density more than 5. We assessed the result of the study shows that the vegetables, basically cauliflower, were highest contaminated vegetable from pesticide, from this heavy metal point of view, and round cod was the least contaminated vegetable from different reasons. And these are the guidelines by WHO, given by the WHO. This should be the level of the heavy metals, but at some places the heavy metal concentration was exceeded from the guideline values of WHO. So important observations are there that manganese was the highest found heavy metal in these vegetables, followed by the iron, zinc and copper. The magnitude of heavy metal detection in different kinds of vegetables was arranged that Vallabhnagar was a site which is very near to the industrial region. So the heavy metal contamination was found highest at the Vallabh Garden and lowest at the Patel Nagar. And if we see the contamination of the heavy metal, then cauliflower was the highest contaminated crop, followed by the okra, then brinjal, then spinach and all. So the effect, you might be aware about the effect of the heavy metals. These heavy metals may cause different kind of the illness, the different kind of the cancers may be created by these heavy metals. And you are aware that one train from Bhatinda to Bikaner, that is known as the cancer train, because Punjab is the region where highest amount of the pesticide is being used in the fields, and the cancer disease cases are very much frequent in the Punjab. So people come to the PBM hospital of Bikaner for the treatment of the cancer, and we are in the process of doing a collaboration with the PBM hospital and the Jodhpur region of Camel Research Center to assess some blood samples of those patients and some camels also, which are nearby to the Raj water treatment plant, the water which is used to grow the fodder for the camels. So we are assessing these kinds of work also. These are the effects of the heavy metals on different part of the body. There is a wastewater treatment plant in the Jodhpur. You can see in this picture, there are used six ponds which are being used to treat the water, but they are just aerate this water and filter this water. The heavy metal do not remove from there. So this water is being used by the camel farm. Behind this you can see some land, there is 400 bigha land which is used to grow the fodder for the camels. So camels' dungs are also studied in our laboratory to assess the heavy metal in their excreta. Some recommendations. Effective wastewater treatment practices must be adopted to minimize the health-related problems. Regular monitoring of heavy metal contamination in vegetable must be carried out. We are also carried out the vegetable samples after 6 months. Every region we are assessing this. Penalties for the industries on untreated discharge of the wastewater. Actually, this is very bad practice in Bikaner region, that people are discharging the wastewater without treating into the field. and you have seen some photographs that sand dunes region, basically the beachwater industrial area is located nearby the sand dunes, and those sand dunes are saturated with that water. So use lake-like structure are there now with the containing this heavy metal, these wastewater. Proper implementation of the acts including Environmental Protection Act 1986, the Water Prevention and Control Act of Control of Pollution Act 1974 are the great need in that region to mitigate such problems. And training program for the farmers should be conducted. We are also awaiting the farmers that not to use this wastewater in that region. And last year we have started a campaign to destroy the crops which are being cultivated, irrigated by the wastewater of the farmers. So district administration has also given us some support to destroy that crop. Thank you very much for listening.
Thank you, Dr. Respect to the time, very nice. Are there any questions? Please. Dr.
Good afternoon, sir. So does it remove only the heavy metals, or because when we remove the heavy metals, does it remove only heavy metals because there are other elements also in the water?
Yeah, definitely. Heavy metals are not only pollutant of the water body, but we are focusing on these heavy metals because the industries which are working in Biyani, they are releasing lots of amount of the metals in their waste. That's why we are focused only on the heavy metals. Otherwise, in our laboratory, different kind of work is going on. Different parameters are being assessed.
Also, the other elements which there are present are good for.
Yeah. See, all these metals which I have shown, the in trace elements, the in trace amount, they are very much useful for us. But if the concentration is increased, that might be create some kind of problems. Thank you. Thank you.
If there are no more questions, I would like to thank Professor Charan for his nice talk. So our next speaker is Dr. Anjali Awasthi. She is assistant professor at the University of Rajasthan, Jaipur. Dr. Awasthi has been working as in the Department of Zoology, and for the last 2 years, her area of interest is evolution of toxicity of nanomaterials. Very important topic in animals and plants. Everybody is talking about the toxicity of nanoparticles, particularly silver, and she is going to talk about the interest in this evaluation. She has got 11 papers to her credit, and one patent registration at Germaplasma PGRC ICR to her credit. I request Dr. Anjali Awasthi. Kindly speak to the time because we are behind schedule over 10 to 15 minutes. Thank you.
Good afternoon, everyone. First of all, I would like to congratulate Biyani Group for organizing such a nice meeting, and especially where the students are interacting. I see so many students asking such nice questions. So again, congratulations to students as well. Myself, Anjali Awasthi from University of Rajasthan. By training, actually, I am a biotechnologist. And recently I am working on nanoparticles, and in collaboration with MIT Jaipur, we are trying to develop a membrane which has anti-fouling properties. When we purify water, then we use certain kind of membranes, and fouling, or especially the biofouling of membranes, it's a complication, a problem. We have to regularly change the membrane, which makes it costly as well. So we are trying to develop membranes so that the fouling property can be reduced using nanoparticles. And one of the case study I will be discussing here, which is on the effect of zinc oxide nanoparticles on biofilm formation in Bacillus subtilis. So for the students, what nanotechnology is, any material ranging between 1 nanometer and 100, which has a diameter between these two sizes, it is nanomaterial. And here in the figure you can see various biomolecules and you can see the range and you can have an idea what the small size of nanomaterial is. Now nowadays why it is important, since in this figure you can see the host is asking, the answer is nanotechnology and what is the question. That means for all the questions existing now, nanotechnology can be an answer. And these are few applications. It is used in electronics, medicines, textiles, cosmetics, transportation, and so many. Endless applications it has in our day-to-day life. We are regularly using the products. Coming to the topic. So, can nanotechnology be used to get safe drinking water? This cartoon depicts. And of course, the answer is probably yes. So we can get safe drinking water, we can remove the problem of membrane fouling using nanoparticles. Coming to water purification systems, the methods we use. So three main methods, the particle filtration, and it filters or blocks few bacteria. Then there is sand particles, yeast, so many things which this particle filtration blocks. Ultrafiltration, of course, which is a better method since it blocks a wide range, and the RO which we use in our home, the Kent RO, AquaGuard, Eureka Fox, the end list. So these RO or the reverse osmosis filters which use membrane, they only allow water molecule, except water molecule, all the molecules are blocked so that we can get purified, safe drinking water. In filters which we use, generally four kinds of membranes are used. Microfiltration membrane having a pore size of 0.1 micron, and it allows viruses, multivalent and monovalent ions together with water. Ultrafiltration, it blocks viruses, bacteria and some solid particles. Nanofilter membrane, it allows only the water molecule and few monovalent ions. And the best one till now, RO membrane, which allows only water, and it has a pore size of around 5 angstrom. Now, what membrane fouling is, which we were discussing earlier. So, this is the new membrane, the new filter which we purchase. Gradually, as the water is filtered, the contaminated water, it has certain sludge molecules, EPS, simple soluble microfluid particles. So gradually the pore size is decreasing, the pores are getting blocked, and finally a thick cake is formed on the pores which make the membrane impermeable, and gradually the water purification is hindered. And this process is called membrane fouling. And this cake formation is due to the formation of biofilm, the EPS secretion by the bacteria, different bacteria which are present in the microbes which are present in the water which we want to get purified. Now membrane fouling, it is in general there are four types: colloidal fouling, organic, scaling and biofouling. So in biofouling process, bacteria, they form the biofilms. Now, why this biofouling is important? Is it really bad for membrane systems? Yes, it is. Why? Because first of all, it reduces the membrane flux. It increases the differential pressure and feed pressure. Membrane is degraded. It increases the salt passage, thereby reducing the quality of purified water. And lastly, it increases the energy consumption. Conventional anti-fouling methods are generally used, which are use of chlorine, chloramines, ozone, iodine, hydrogen peroxide, formaldehyde, butyraldehyde, etc. We are focusing on we are trying to develop functionalized membranes using nanoparticles. So for that particular study, we have used zinc oxide nanoparticles which were synthesized by chemical wet method, and they were characterized using XRD, UV-vis spectrophotometer and TEM, which says the size was hexagonal and the shape was hexagonal and the size was on an average around 14 nanometer, which was cleared by XRD also. Now, the next step was to check the antibacterial activity of these nanoparticles on Bacillus subtilis. So we used wet diffusion and disk diffusion method, and we found after keeping the bacteria overnight using nutrient agar media that the growth of bacteria was inhibited in the presence of zinc oxide nanoparticles. This was confirmed, and we want to check whether the nanoparticle is killing the bacteria or it is inhibiting the growth of bacteria. So we use 2-difixy and PI, propylium iodide, and this is the normal. Here I think due to the problem of projector, the red color is not that much clear. So this is the control where all green cells are observed, no killing, and then gradually the red cells, these are the cells which are getting killed, the bacterial cells, the killed ones, and the green, they are the live cells. So ZNO nanoparticles, they are inhibiting as well as killing the bacterial cells at different concentrations. This is the SEM image. In control, we can clearly see a good amount of bacteria, but when they are treated with ZNO nanoparticles, the shape and the size of bacteria is getting changed. They are clumped, their membrane is damaged, and they are round in shape as compared to the control one. Now coming to check the biofilm activity of bacteria, we incubated the bacteria. These are the control, and this is the broth with nanoparticle. After 24 hours of incubation, we check, and in control you can see the white colored biofilm formed, which is adhered to the test tube wall, and this biofilm it is reduced in the treated samples. To further check a thick band, crystal violet stains the polysaccharide present in the biofilm. So a thick band of biofilm we can visualize, and which is reduced in the treated samples. Now, in order to quantify, we dissolved the crystal violet in acetic acid, 30% acetic acid, and we kept it for incubation around 10 to 15 minutes, and then we used spectrophotometer to get the readings. And here you can see this is the acetic acid, the plant control, dark blue color due to the presence good amount of biofilm, and the color is reduced. That means the biofilm formation is reduced in the treated samples. Okay. Then we check the micromorphology of biofilm using SEM. Here in this figure you can see thick layer of biofilm, and here the black arrow shows the adhered cells, the red arrow shows the biofilm. This is the control one, and in treated one there is reduced or negligible amount of biofilm formed, though both the plates were kept at same time of incubation. On further magnification of the same slide, we can see that there was a linear chain formation, which is the initial stage of biofilm formation in treated one, whereas the number of cells are higher. Here the number of cells are reduced, and you can see the biofilm as well. On further magnification, we can see the round cells, the damaged cells, the membrane is damaged, deformed, and here also similar results. we can see the nanoparticles also, which are there to the bacillus cells. Now, this ZNO nanoparticle, why it is inhibiting or killing the cells? So, some oxidative stress it may be inducing in the bacterial cell, due to which the cells are not normal. So, in order to evaluate the oxidative stress, we use Catalase Assay. The control one, this is before incubation, and then at the end we can see together with Catalase, what Catalase does, when we add hydrogen peroxide, it releases oxygen, and this oxygen was made to bind with Triton X100. So this Triton X100, it captures the oxygen released by the H2O, sorry. And this layer of foam formed, it is the control one, a thick layer, that means high level of catalase activity, and gradually this foam is getting reduced, which clearly indicates that catalase activity is also getting inhibited. Now at the end, I would like to conclude that ZNO nanoparticles, they are affecting the activity of biofilm in bacillus. Around 86% production was observed, morphology was changed, and now based on these results, we are about to fabricate membranes with nanoparticles of ZNO, and we will check the biofouling activity. I would at the end acknowledge my students Pooja and Arpita, Soft Materials Lab, MNIT and DST Government of India for providing funds. And thank you all for patiently listening. The question is open for questions.
Thank you, Dr. For a wonderful talk on the prevention of biofilm using the oxide nanoparticles. The talk is open for questions. Any questions?
Excuse me, ma'am. How do you measure biofouling activity?
Actually, right now we have not, I told you that this is one of the case studies. Right now we have only checked the antibacterial activity and the biofilm activity, not the anti-fouling activity. We are in the process.
Is there any process to measure that?
Yes, there are. Like, first of all, what we will be doing, like we are using the same nanoparticles, ZNO nanoparticles, and we are functionalizing it, and we are making it to get entered in the pores of the membranes, and then we will be checking the activity.
But fouling is smell. So how will you detect that smell?
This anti-fouling actually is the blocking of the pores of the membrane. So, and the bacteria is getting developed over there, so we get the smell. So if we are reducing the biofilm activity, that means the fouling activity, anti-fouling activity is.
Thank you. Good afternoon, ma'am. And my question is that if the filters only allow water molecules to pass through it, then is it useful for us?
We want to get purified water. So, but we have some questions when we go to purchase an RO. Like RO water, the RO filters, they remove all the minerals. We have the problem. Like we are not getting minerals from the water, then it may be harmful. But we don't get minerals only from water. We use vegetables, fruits, different food products to get the minerals. So it is okay. We can use.
Thank you.
So, any questions from this side? All the time there are questions from this side. One last question, please.
Good afternoon, ma'am. Why nanoparticles are given so much of preference? Can we use other metallic oxide nanoparticles?
Of course we can use, but I'm working on ZNO and they are widely used and they are cheaper as well as compared to other oxides and easy to get synthesized in the lab. So we are using it.
Thank you.
Thank you. Thank you. Our next speaker is Dr. Vishnath Tiwari. Dr. Vishnath Tiwari is from Central University, Rajasthan. He received his PhD from College of Medical Science, New Delhi on membrane proteomics. Presently, he is assistant professor and coordinator of Department of Microchemistry at Central University, Rajasthan. His areas of interest include proteomics, mainly membrane and differential proteomics, biochemical studies of the proteins, and in silico protein-drug interactions, which is associated also of journal integrated OMICS and bioinfo journal of proteins. It is also an editorial board of members of the variant to the Journal of Medical Society, Biology, Journal of Biomolecular Structure and Dynamics, International Journal of Applied Biotechnology, Biochemistry, Technology, BICON, Biochemistry International. Congratulations for handling all these journals. I request Dr. Vishnu Tiwari to bear this talk.
I would like to thank organizer for giving me this opportunity. Topic of my presentation is multidisciplinary approach to understand cardiovascular resistance. I have taken a model that is escape pathogen and a bacteria that is Acinetobacter baumannii. As you know that in bacteria that causes nosocomial infection means especially the hospital-acquired infections for a student. So there is a group of pathogens known as escape pathogen. Out of them, Acinetobacter baumannii is one of them that causes the nosocomial infection. And report shows that people are now more dying as compared to HIV and tuberculosis combined. So Acinetobacter baumannii is reported as a threat to the soldier wounded during military operations in Iraq as well as it is also isolated from the natural resources. As I told that this Acinetobacter baumannii is going to be found in the hospital and we are using many disinfectants like ethanol and other to clean the hospital setup. But interestingly, Acinetobacter baumannii is going to use these molecules as a carbon source, as an energy source, so that they have better survival. So whenever we are giving any antibiotic to any individual, then antibiotic must cross the membrane and then it will enter into the pathogen. As you can see that the membrane consists of three layers. We have outer membrane, we have periplasmic space, and we have inner membrane. So whenever we are giving antibiotic, the antibiotic must enter to this membrane and then it will inhibit the bacteria. So I have taken a model that is carbapenem, which is generally prescribed antibiotic for Acinetobacter baumannii, and you can see that beta-lactam antibiotics. This is carbapenem is an example of beta-lactam antibiotics. It will enter and bind to the penicillin binding protein, which is well known for its involvement in the cell wall biosynthesis. So when a beta-lactam antibiotic is there, it blocks the cell wall biosynthesis that will lead to the death of the bacteria. And beta-lactam group of antibiotics are mainly classified into the four groups: penicillin, cephalosporin, monobactam, and carbapenem. And carbapenem is a recent one. But the resistance developed against the carbapenem is important here, and it is going to lead to the significant health problem, and it is associated with high morbidity and mortality. So I am telling you what is my experimental approach to find out the resistance mechanism in the strain we have collected. So firstly we have collected strain for carbapenem resistance using MIC and electron microscopy. Then we have performed, as I told, that membrane is playing significant role in transport of antibiotics from outside to inside the bacteria. So we have performed the proteomics of the membrane to find out the protein which is involved in the resistance. And then we have also purified the specific protein which is differentially expressed in resistant strain and sensitive strain. And then we have characterized it by using biochemical and biophysical approach. And then in the last we have tried to find out the probable solution of this antibiotic resistant strain. So we have collected these 29 strains from all India Institute of Medical Sciences, and it was found that most of the strain which is collected is going to be effective when we have then combination of antibiotics given. But because our aim is to find out the carbapenem resistance mechanism, so we have identified the MIC of the collected strain for the carbapenem, and we found that about 35% of the collected strain are resistant to the imipenem. So we wanted to know, is the imipenem is going to have effect on the membrane as it is going to inhibit the cell wall biosynthesis. So we have selected one strain, that is RS307, is is a representative of resistance state, and one type state, that is ATCC. So we have performed the TEM, that is going to give you the membrane structure. So you can see that when the sensitive stain is treated with the MA-penem, you can see the bacteria is going to lyse. But resistant stain after treatment with the MA-penem is going to form osmotically stable state. That it shows that the resistant stain must have something which basically makes the pathogen survival in the antibiotic pressure. So we have performed the proteomics. For that we have isolated the membrane proteins from sensitive stain, from resistant stain. And we have used the dyes approach. Well, what is there in the dyes approach? I am telling you the one model of it. So one protein isolated from resistant stain is labeled with the fluorescent dicyclate. Protein isolated from the sensitive stain is going to be labeled with the Cy5, and then we have mixed it and perform a dyes experiment. Okay. So in the dyes experiment, we have because of shortage of time, I am just briefing it. But dyes experiment shows that we have found some overexpressed protein only in the resistant stain that was not there in the sensitive stain, and we have also found some protein which is downregulated, which is present in the sensitive stain but not found in the resistant stain. So we have taken that protein out and then perform LCMS analysis to identify what are the proteins which are overexpressed in the resistant stain, because they are going to play a role in the resistance. And this is the mass result. So we have concluded that when we have sensitive stain, beta-lactam antibiotics enter and bind to the PBPs and inhibit the cell wall biosynthesis, and that leads to the death of the organism. But resistant stain overexpress many proteins. Out of them, OXA51 beta-lactamases, MC beta-lactamases, and it was also found that the protein which is involved in the metabolism is also upregulated. So that means we can say that from the proteomics result that these are the proteins which are overexpressed in the cell resistance state, and that might play some role in the resistance of the resistance state. But until we will not validate the particular protein, we cannot say, because proteomics only explains that we have overexpression or downexpression of the protein. So it is important that every protein must be validated for its function. So we have selected many proteins, but I am presenting only one protein is here, that is OXA51, which is overexpressed in the resistance state. So we have cloned OXA51, and as you know, the sensitive state is the one which is going to be killed by the beta-lactam. But when we have transformed OXA51 in the sensitive state, we have found that OXA51 is going to give resistance to the sensitive state. So it is proved that the OXA51 might have role in the resistance. So we have purified the OXA51, and then we have performed this biochemical and biophysical analysis. And biochemical and biophysical analysis shows that this OXA51 is going to have good affinity for carbapenem, that is imipenem is a model of it. And then we have performed the stability of it, because when you are talking about the survival of resistant stain into the hospital setup, the protein must also have some resistance to the pH and temperature. And it results shows that the secondary structure of OXA51 is going to be resistance with change in the temperature, as well as it is so resistant with the change in the pH. And so we also wanted to know what are the molecular interactions that is involved in the interaction of OXA51 with the MA-penem. So we have modeled OXA51, and then we have performed the in silico interaction studies by two independent software, Klein and Gold. Both are using different algorithms. One is based on energy, another is based on genetic optimization. And results shows that the OXA51 is, we have biochemically proved it, they have having interaction, we have biophysically proved it, they have survival in the adverse situation, and now we have also proved that they have better interaction with the antibiotics. And then we have also performed the simulation study of imipenem with the OXA51, and we have found that throughout the simulation the imipenem is present inside the active site of the OXA51, which shows that it is going to have better substrate for OXA51. And then we have also performed the proteomics of outer membrane, and the outer membrane proteomics result shows that the resistance strain overexpressed efflux pump. It's alter the OMP protein which is involved in the influx of antibiotics. It overexpressed iron binding protein, because iron is important for the survival of resistant strain. and it also alters the CARO because CARO is a channel for the entry of antibiotic inside the host. Similarly, we have also performed the proteomics in presence of iron and absence of iron, because what happens when pathogen infects the individual, it basically sequesters the iron. So there is a fighting of between organism, pathogen and host for the iron. So who wins? If pathogen wins, it will acquire iron. If we win, we will basically kill the pathogen. So we have performed proteomics and the result shows that the resistance strain is going to overexpress 5 receptors for iron acquisition, and that is ferric, acetobacterium, ferrin, and then few receptors. They all are involved in the sequestering the iron from the host. So we know that from this discussion, we know that the resistance strain is going to be more rigid to the pH change, more rigid to the temperature change. It is going to have developed protein which is going to survive in the presence of antibiotic by cleaving it, and it is also acquiring the mechanism which is going to basically survive better in the host. Then what is the solution of it? So we have tried 2 approaches which can be an alternative of the carbapenem. So first approach is going to be the nanomaterial based approach. So we have synthesizes silver nanomaterial, and I am telling you the silver nanomaterial is going to be antimicrobial activity for many pathogens, but we wanted to know if we enhance the specificity as well as antimicrobial activity of the silver nanomaterial, what we can do. So we have selected 5 capping agents. So what is the significance of capping? When we cap the silver nanomaterial, it will basically stabilize the silver nanomaterial inside the host, so it have more antimicrobial activity. So we have selected 4 nanomaterial, and we have found that the PVP cap silver nanomaterial shows antimicrobial activity against the resistance state. So we have only proved at this particular time that PVP cap silver nanomaterial is going to be a oxide. But what is the mechanism of PVP cap silver nanomaterial? We don't know. So our next plan is we are planning to perform the proteomics of resistant strain in the absence and presence of the PVP cap silver nanomaterial. So when we found the result, we might basically know what is the mechanism of action of this PVP silver nanomaterial. So similarly, we have tried the other approach as a herbal active, searching the herbal active compound which is going to be effective against the carbapenem resistant strain, and we have found that the polar extract of two plants, that is bale and madar, is going to have some antimicrobial activity. So our next aim is to make a fusion of active compound of this plant. Although it is just a preliminary screening, we have to perform HPLC, and then we have to screen out the molecule from the plant, and then we will make the fusion of silver nanomaterial by using that particular active molecule, and then we might find the solution of this carbapenem resistance. So the conclusion of this study is we can use multidisciplinary approach. As you can see that we have used proteomics, we have used biophysics, we have used bioinformatics, we have used nanotechnology to find out understand the carbapenem resistance state, as well as we have to find out the solution. So these are the 16 publications which I have mentioned here. Although I am just putting everything very quick way, so you might not understand. I just briefly tell you. So this plant, the work which is basically screening of a particular plant for carbapenem resistance state is published in microbial drug resistance, and the result of the proteomics which I have mentioned is published in the Journal of Proteomics, Journal of Proteome Research, PLOS 1, and then other biophysical work is published in JBSD, and so on. So I would like to acknowledge Professor Rajeshwari, Dr. R.T. Kapil, Dr. Subra Rao from AIMS, and then from JNU for giving me strain as well as proteomics facility. I would like to thank my student who is working in my lab to make everything possible. Thank you.
Thank you, Dr. Biyani, for the excellent talk and a lot of published work in the proteomics. The talk is open for questions. Any questions? And students?
Yeah, if you see that one receptor we have found that is basically present in the resistance state is a receptor. So when resistance state is there inside the host, it will release cytophores to sequester the iron. But it must also have some receptor which will bind to the cytophore. So the resistance state secreted cytophore, it will bind to the iron inside the host, and then we have the cytophore receptor present in the resistance state, so it will basically sequester it and take it for its metabolism. So we have found the cytophore overexpressed as well as cytophore receptor is also overexpressed in the resistance.
Any more questions? No more questions. So kindly join me for the Applause for Biyani. We have the last part of the session by Dr. Deepak Sharma. He also comes from German Life Post, and Dr. Sharma is an associate professor, Department of Science, Biyani College of Economics, and also holds a position of research scientist at Biyani Biosynthesis Biosolutions Private. He has done his PhD from University of Milan, Italy, followed by the postdoctoral stay in the European Union. His research areas focused on nano-bio-interface, investigating cell-substrate interactions, biocompatible materials for tissue engineering, and nanobiosensors for diagnostics, real-time health monitoring, and he holds a patent and has published peer-reviewed publications in the area of nanobiotechnology. Dr. Could you mention about this?
Good afternoon to you all. Today I am going to talk about the nanobio interface regulates cell-substrate interaction. Before going to the nanobio interface, it's better to have a Before going to the nanobio interface, it's better to have an idea about what is the nanotechnology and do we really need it. As map already told, nanoscale varies from 10 to the power 4 to 5, which is the cellular dimension, and scale from 1 to 100 nanometer. 1 to 100 nanometer, that is the dimension for the proteins, DNA, and viruses. So basically, when the DNA was discovered in 1950s, so we got an idea to develop a kind of system that can conjugate with the molecules and we can extrapolate the biological information into the in vitro system. Since after 1950s, we got the first work in 1990s when Professor Ijiyama from Japan successfully developed the carbon nanotubes. So we first got the nanostructure in 1990s. Before that, we were just hoping to have a material that can integrate with the biological system to have the information in the in vitro studies. After Ijiyama in 1993, in 2004, first we got the information. First we got the material, another material that is graphene, developed by Professor Dean from University of Manchester. And that was basically the same molecule, just they opened the structure of the carbon nanotube and it was just sp2 hybridized, just a carbon sheet. So both these nanomaterials, they have extraordinary properties, they have extraordinary chemical, physical, chemical, electronical properties, and because of that only we have an opportunity to use these materials to mimic the biological system to develop a device for the diagnostics and for the therapeutics. So now, what is a nanobio interface? So we have an idea about the nanomaterials, we have an idea about the biological system. So when the nanomaterials, basically the particles, the graphene sheets, or the nanotubes, when they interact with the biological system, they have a very small area that is called as, that area is in microns or in nanometers, and that area is called as a nanobio interface. So this dynamic area is coming with many challenges. Because still from development of carbon nanotubes in 1993 and after that in 2004 the graphene, still we are waiting for a commercial product that is totally based on the nanobiotechnology. So it means this nanobio interface comes with many challenges and also some limitations that we cannot regulate the properties, the dynamic area, the dynamic region that is called as the nanobio interface. Basically, it comprises the region that the matrix faces towards the nanoparticle, the matrix itself, and the matrix faces towards the cells. So, as you can see here in another picture, when the nanoparticles just enter inside the biological system, There are the various proteins, lipids, and glycosylated molecules. Those molecules, they are charged, and the surface of the particle is also very active because the surface to volume ratio increases when the size of the nanoparticle decreases. So because of the active surface of the nanoparticle, these molecules, they just got absorbed on the surface of the particle. The first challenge arises here. We have to specify the molecules that can absorb on the particle. So after the absorption of the particular molecule on the surface, these molecules, they just they just move towards the target and they just move towards the cell membrane. And these cell membranes, they have the specific receptors on the surface. These receptors, they just bind to the molecule that is loaded with the biological sensors. These biological sensors, they just transmit the information and initiate a signaling cascade, and that cascade is very, very important to understand, to mimic the information that occurs naturally inside the system to develop a device or diagnostics. So basically I work in this area that is nanobiotechnology, and my aim is to develop a device and various materials digitizing biological information electronically. So basically there are a range of nanomaterials, the carbon nanotubes, graphene sheets, and different kind of nanomaterials, basically polymeric or metallic nanoparticles. So having the information from these nanoparticles, taking the control from the biological system and put everything on the biological chip onto the electronic system, so that we can just have the information that is happening inside naturally. It will help us to develop a device or diagnostics. So there are a range of techniques to have this nanotopographical surfaces. Basically, we use the physical techniques and we use also the chemical processes to have these approaches to have the surface to mimic the biological system inside, that is the cells. So basically it initiates with the microscale topography. After that the submicroscale topography comes, and when the cells, they basically interact with the surface, they have the specific receptors on the surface, and those receptors are called as integrins. They connect with the external environment, they connect with the external surface, initiate the signaling processes inside the cell, and cell itself gives us the information where we have to go, where we have to proceed. Keeping in mind all the cues, we developed a kind of system that is having the different topographies on the surface. So basically the idea was to develop to have a system with the different thicknesses. So we deposited different thicknesses by using a pulse cluster microplasma deposition operators, and from the left to right we can see here that it is having basically a very darker area. This darker area is having a high thickness area, and as we move towards the right we have a very low thickness area, and this corresponds to the roughness. As we have a table here, we can see that the 50 nanometer thickness corresponds to a particular roughness, that is 14 point something. As we increasing the thickness of the material, we are increasing the roughness of the material. So by this way we are controlling the roughness of the material just by regulating the thickness of the sample. After that, it was first very important to understand whether the thickness is stable or whether the sample is not oxidizing. So we first performed the AFM studies, and we found that as we are increasing the thickness, the grains, the nanomaterials grains, basically they are large in number, large in size, as well as the pore size between the grains, they are increasing. So we concluded that the material is not oxidizing when it comes into the air, and we can stably deposit a particular thickness that will conclude to a particular roughness. After having the initial the results, the next was to have the information about the chemical properties of the material. We used titanium in this case, and we found that after heating up at a particular temperature, the titanium oxidizes to titanium oxide. So another question comes, if we are using a thin film and that thin film is just for the titanium, or if it is made from titanium oxide. The property of the material changes, and it controls also the behavior of the cells. So we find out that after heating at a particular temperature, we find out the presence of the titanium only. It doesn't oxidize at the temperature, and it doesn't transfer into the another phase of the material. Then we performed the scanning electron microscope studies, and we found that the material was also stable and it doesn't disintegrate into grains. And as you can also see that at a higher roughness, the grains are larger and the pore size is also larger, and as a lower roughness, you can see that the sizes grains are also smaller. After having the material, we moved to the cells, and initially we took the neuronal cancer line cells, because the neuronal cells, they are the most sensitive cells. And we find out that at the lower temperature, at the lower roughness, the cells are feeling pretty happy rather than moving to the high roughness. So we did the study for the 72 hours, and we find out the lower roughness surface provides the most compatible environment for the cells to grow. Here we find out the first platform to help to develop a device that we can culture the cells for the 72 hours under a stable environment that can give us the information for the diagnostics. After that, we move to the immunofluorescence studies, and we find out at the lower, the resolution is not so clear. At the lower roughness, the neurons, they are differentiating properly and they are forming the neurites. As well as we did the vinculin studies, it shows that the focal adhesion of the cells. Focal adhesion is basically demonstrate how firmly the cells they are adhere on the surface. So we calculated the length of the focal adhesion, and we find out that the lower roughness is crucially controlling, regulating the behavior of the cells. As we are increasing the roughness of the surface, the cells are not compatible with the environment and they are dying. So after having the initial information that only the nanotopography is crucial and controlling the cellular behavior, it was very crucial to understand whether the nanochemistry, as I demonstrated before, the nanobio interface, it doesn't only have the nanotopography, it also has the nanochemistry, the biological matrix where it combines. So we have to develop a kind of system that allows us to think that whether the nanochemistry and nanotopography together crucially controlling the behavior of the cells under in vitro conditions. So we developed the system by using initially we deposited the different thicknesses on the sample, the different thicknesses, and after that we deposited a different 5 nanometer layer by using the different instrument just to have the different chemistry on the surface. And after that we developed a kind of microarray. This microarray was having 50 to 500 nanometer thickness, 15 to 30 nanometer roughness, 162 total areas of the diagnostics, and 54 different compositions having metals and topographies. So 9 metals were used to develop a kind of system, and we developed the microarray that was having the different topographical regions as well as the different chemical regions. After that, we used the studies, compatibility, topography, surface chemistry, and microscopic analysis to evaluate our results. Initially was when we are depositing another metal on top of the roughness, on top of the topographical substrate which we have. So our first aim was to have that these 5 nanometers layers, these are not changing the initial roughness of the material. So we find out that after depositing another material, it is not changing the roughness of the total initially deposited titanium. So our aim was achieved in such a way that we can change the chemistry of the surface rather changing the topography of the surface. We again perform the AFM studies to have the granular structure, that whether if we are depositing another 5 nanometer layer, whether it is changing the granular structure of the film or not, and we find out that it is not changing the granular structure of the film after depositing by the plasma deposition. In the end, we perform the cell culture, and we use the same neuronal cancer cell line, and we find out that the particular metal at a particular roughness is very crucial to regulate the interaction. So, as we can see here, the gold is the best material that can interact with the cells. They are much more compatible at the gold surface. After that, the aluminium and copper, they are almost compatible with the cellular behavior. And as we can see here, the roughness, as we are increasing the roughness, the most materials that are chromium, silver, and zinc, they are not that much compatible with the cell line. So we can conclude here that in the end we applied for a patent, and we developed a device on base of this technology that Initially it can be used for the detection of the cancer, that based on the gold nanothin film at a lower roughness. So these different type of materials demonstrate that the particular material, the nanochemistry is equally responsible, equally regulating the as the nanotopography. Then we did the calcium staining for the viability of the cells, and also the cellular behavior was studied with different immunofluorescence molecules, and we saw that again the same results that are getting before with the DAPI studies. And I thank you for.
Thank you very much for the nice talk. Talk is open for discussion. Couple of questions more. No more questions? If there are no more questions, kindly join me for applause to Dr. Vimal Sharma. With this, we come to the end of the session. I would like to hand over the session to the organizers. Thank you for giving me this opportunity to chair this session.
Thank you so much, sir. Wonderful session we had with our all eminent speakers. So we would like to show our humble gratitude towards them for giving their valuable time to us today. So once again, big round of applause for our all eminent speakers. And we would also like to pay our humble and honored gratitude to Professor Manohar Badekar, sir, for handling the session very beautifully. Sir, your presence was very energetic and interactive. So once again, thank you so much. Please big round of applause for Professor Manohar Badekar, sir.
Now is the time to honor our speakers. So I would like to invite Professor Biyani, sir, to give mementos to our speakers. So firstly, I would like to request Professor Biyani, sir, to give memento to Dr. Maiko Akajima. Please, ma'am, come on the stage. To give a huge round of applause for the ma'am. Thank you, ma'am. Now I would like to request sir to give memento to Dr. P.V. Charan, sir. Thank you, sir. Now I would like to request to present memento to Dr. Anjali Avasti, ma'am. Request to present memento to Dr. Vishwanath Tiwari, sir. I would like to request to present memento to Dr. Vimal Sharma, sir. Now is the time for tea break, and along with this there will be poster session 2, and all the candidates for the poster presentation are requested to assemble in tea area, and we will come at sharp 4:30.